Many of you would be aware that we've developed a network called the Pancreatic Cancer Network. This is specifically because the volume of patients with pancreatic diseases at any one institution is not significantly high. And whilst we can develop a specialist centre for colorectal diseases, say at each individual hospital, it's very difficult to do that with a disease prevalence which is lower. And so we developed the New South Wales Pancreatic Cancer Network to be able to provide a virtual network where people with expertise and interest in the area can come together and also liaise directly, provide a support network for patients, and also provide a basis for ongoing research. And bringing this together has been relatively successful, and it's really bringing us to the forefront of research for pancreatic cancer globally in the future. Most recently, as part of the International Cancer Genomes Consortium, which is a consortium of countries which has come together to sequence 50 of the most common cancer types and subtypes. Australia's cancer is pancreatic cancer, and we're starting to work on that at the moment.
We all recognise that unfortunately pancreas cancer has proven a very difficult target for myriad of reasons, from the intrinsic genetic change, the fact that it's often discovered very late, when it's very advanced and inoperable, to maybe some unusual features of subversion of the surrounding stroma, in that pancreatic cancer appears to have very well developed mechanisms for subverting the stroma to increase both the neoplastic potential and the metastatic potential. Nevertheless, as frustrating as it is, we have made some small incremental gains. There are patients who are living longer. They're not living years longer, maybe months longer, but they're certainly living longer and living better. And we are getting, all of us are getting the occasional patients living surprisingly beyond any of our expectations.
When Mum was first diagnosed, she told me that the doctor said she would only live for about 2 weeks. And as it turned out, Mum lived for 3.5 years, and she lived pretty well for most of it. She had a reasonable quality of life.
So there are clearly clues to the future. And as you all know, the revolution in genetics and biology has finally hit the clinic. And this is just as true of pancreas cancer as it is for others like colon cancer, that we are unlocking the keys to particular targets and thereby identifying novel targeted therapies to add to our standard treatments. We're in the middle of a growth phase in pancreas cancer in terms of genetic understanding, and although there have been some acknowledged setbacks in targeted therapies that have not translated into a change when tested in the clinic, certainly there have been some small gains and at least some directions to go in. So what we really need now are 2 very important initiatives. The first is we simply have to get better at collecting tissue from our patients. Until we get decent tissue banks, we're not going to be able to really properly identify the targets. Pancreas cancer is a difficult one. Often it's fine needle aspirate biopsies that we use that is very limited material. We simply have to get better at collecting decent tissue specimens from the people in whom we can collect it, and make a determined effort for those who are lucky enough to be able to have a resection, or at least an open procedure to see if they're resectable, to making sure we collect tissue from those patients so we can start to interrogate it and get some new directions. And the second, of course, is participation in trials of novel targeted therapies that are informed from tissue collection and gene array studies. Where are we headed at the moment? It's just like every other cancer. We're hoping that some forms of targeted therapy will prove to be useful in the clinic, and the only way to do that is to do the clinical trials, because repeatedly we have seen that the mouse models and the in vitro models can only take you so far, and there still is no substitute for the trials in humans.
Dad went on a trial, and he was on the control arm, so it was never about getting some sort of massive therapeutic gain. Can't tell you how valuable that was. It managed everybody's stress. All of a sudden you have people with an extra fuss factor. So you go and you see the same nurse every time you go through the hospital system, and they take extra notes of you. There's cab charges to get dad home from the hospital, so mum's not sitting there waiting while he has chemo. He can, do it himself. So there's that whole independence factor.
It's probably even more important in pancreas cancer than it is in most of our other more common cancers to really encourage our patients to participate in clinical trials. We have major hurdles to overcome, and it's only by over-recruiting to clinical trials in pancreatic cancer that we can start to make the sort of progress that our colleagues are now making in colon and breast cancer, and even to a lesser extent lung cancer, and try and move the field forward.
One of the exciting things for me is that I've become involved in the Colling Institute, where we have some wonderful scientists working with proteins to try and find new biomarkers for pancreatic cancer. The biomarkers that we're interested in will be a group of proteins in the blood which help to, one, make the diagnosis, and two, help to give a prognosis of outcome.
The drugs that we use for pancreatic cancer at the moment don't work equally in all patients. They work better in some patients. They work very well in a small group of patients. We only have a few to choose from, but we want to be able to make the choice as rapidly as possible and put them on the right drug first. And we can do that by understanding more about the biology of the cancer, by understanding the genetic abnormalities that underpin a particular subtype of pancreatic cancer based on their biology, and then put them on the most appropriate therapies as rapidly as possible. So our overall goal with the research we're doing at the network, the Garvan Institute and at Bankstown Hospital, really is to develop personalised therapies for pancreatic cancer. Certainly we know that surgery doesn't work for all people, but there are some instances where we should be operating on people when we don't, and there are some instances where we shouldn't be operating on patients because we're not going to do them a great service. And so we can start using molecular markers, biomarkers, to help us guide some of those treatment decisions, and we're currently trialling some of those into the future.